Welcome friends. Now we will discuss another special type of embryonic development that we call the apomixis. Apomixis, we call it formation of seed without fertilization. That is the alternative mechanism to sexual reproduction. So usually in a plant's reproduction, three types. When you discuss reproduction, it may be asexual or maybe sexual. In asexual reproduction, a part of the plant, vegetative cells have containing more than vegetative cells. This part may develop into new plant. Different types of asexual reproductive structures are produced like tubers, rhizomes, corms, etc. Sometimes leaves are also produced new. These are the asexual reproduction. And in sexual reproduction, what is the formation of seed, where the male and female gametes fuse is forming the zygote developed into the embryo, and the ovule developed into the seed, and ovary into the fruit. There is a common process of sexual reproduction. But sometimes there is a special type of sexual reproduction, we call it selfing sexual. The third type reproduction, where even there is no sexual union or sexual fusion of male and female gamete resulting in the zygote, still the embryo can be formed or seed can be formed. The formation of seed that occurs without fertilization, there is no fertilization. So this was first also reported the same case. This apomixis and polyembryony both are of similar cases, same case. When in a polyembryony the embryo forms the vegetative part without fertilization, that is apomixis also. But poly means many. In apomixis there may be one embryo or maybe many embryo, but apomixis is a special case where only the seeds are contain the vegetative embryo, not the sexual embryo. That is the reason they are called apomixis. So there, this was also first reported by the same person, your Leeuwenhoek reported, and later this apomixis character was also found in many other plants. So this further classified may gametopathy. As you know, in a angiospermic plant, this is the ovule, this is the integument, this is the nucellus, this is the embryo or the egg, here the synergids or the antipodals, like that, this structure. So here in case of apomixis, from where the cell is, which cell develop into embryo, which cell develop into embryo of the seed. When there is a seed, means embryo must be there. In a seed, first thing is embryo, then other things are there. So when which is cell of the total ovule forms the embryo without fertilization, accordingly they are classified. It may develop from this gametophyte tissue, it is gametophyte tomorrow, haploid tissue. Gametophyte tissue जदि हैला ताको तमर कहला कोन ना तमर gametophytic polyembryony. जदि sporophytic tissue, your nucellus and this integument सेठु जदि embryo formation हैला ताको कहला तमर sporophytic polyembryony. Accordingly, it is also called the adventitive. Sporophytic also called the adventitive polyembryony. From the other part, like adventitive root, because normally the embryo should be from the gametophytic part, but when it is from the sporophytic part, we call the adventitive polyembryony also. The sporophytic also called that. So here what happens in gametophytic polyembryony, it may be various types. It may be, as you found, apospory and diplospory. So apospory means somatic cell forms the embryo. Apospory means automatically spore forms, spory. So this somatic cell may develop into the embryo sac. Here embryo sac is formed, but there is no fertilization. So any somatic cell of the gametophyte, another other cell here, it may develop into the embryo sac. And in case of diplospory, there is a diploid embryo sac. The embryo sac, what happens? The embryo sac actually it is haploid because of the formation of your microspore. So megaspore. Megaspore is haploid because of the development of during sporogenesis. The megaspore mother cell form the haploid megaspore by meiosis. But sometimes what happens, the mitotic megaspores are produced. These mitotic megaspores are resulted to mitosis. Mitosis is a megaspore diploid megaspore. In that case, a diploid megaspore will develop into the diploid structure, which will form the embryo, that will call the diplospory. And this diplospory sometimes is mitotic or sometimes is meiotic. In meiotic diplospory what happens? Normally what happens, this microspore forms because of the meiosis. Meiosis occurs. this microspore mother cell or MMC undergoes meiosis forming the microspore. But sometimes during meiosis it becomes abortive. Meiosis battery other bond here because abortive, and again the mitotic starts. So that the in a meiosis what happens? It divided into just chromosome sets. It becomes two equal half sets. So N and N. So what happens when N and N they separate? Here it breaks. It becomes the meiosis becomes abortive. So that both the haploid sets remain in the same nucleus, and then mitosis starts. So meiosis actually happens, but after meiosis that is become abortive, so that you find diploid meiotic or dimeiotic cells we call them. The diploid cells are there. So because of this we find this meiotic apomixis or formation of the seed. And in the case of mitotic what happens? Instead of meiosis, there is no meiosis. Megaspore mother cell undergo mitosis forming diploid megaspores. Megaspores having 2n number of chromosomes. Here there is a meiosis then and they are abortive, but here is a mitosis. This is about the apospore and diplospore. Now other cases when the sporophytic tissue there is nuclear integument. Sometimes the nuclear tissue, nuclear cells they become active from the embryo sac. Nuclear cell टा diploid cell. So diploid cell active for some reason active हो जाउची जा फले से formation करछि कोन गोटे embryo sac formation करछि. It pushed into this vicinity and becomes the embryo sac. So here this seed is formed and this contains the embryo, and this is because the nuclear, this is nuclear apomixis. And very rarely the integumental cells, the outside cells covering the ovula, covering the what is outer inner integument, right? Because there are two integuments, testa and integument. The outer or inner integument cells, mostly inner cells, they become active. They divide rapidly forming the embryonic structure that remains in that seed. So these are the different types of your apomixis, which may be gametophytic when developed from the gametophyte, which may be sporophytic when developed from the nucellus or from the integument. In case of sporophytic, they are called adventative because they are not subjected from the sporophyte. Usually कौन होगा ना तमरा जो एम्ब्रियोटा फॉर्मेशन में gametophyte होगा, gametophyte sporophyte आइसो. There is जो जाइडो sporophyte हे गला गोटे जेनेरेशन ओमिट हे गला. So disadvantage. Advantage जाते कौन ना गोटे जेनेरेशन तो फ्यूजन होउची मेल और फीमेल गैमेट, we find new characters. There is vigority of the plant is restored. यूनियन होची मेल फीमेल सीडटा हला तार vigority restore होची. जदि न हबो तार vigorटा growing capacity टा कमी जिवो. Vigority of the seed is not restored here. When they develop directly from the nucellus or the integument. But anyway, that is the adventative found in many plants. This is adventative apomixis, where the cells of the sporophyte of tissue of the ovule, they may develop into embryo. These are different types of apomixis found in angiospermic plant. And you see, in case of apomixis, it is not found in gymnosperms. Polyembryon is a common character in gymnosperm, more common in gymnosperm. Gymnosperm में polyembryon होउची. Difference कौन? Polyembryon में more than one embryo. Apomixis रे कौन? Embryo टा होउची without fertilization. There is one embryo, maybe more, but usually one embryo is formed. But there are different pathways. There are different pathways to form the embryo, not due to fertilization. So this is the difference. So apomixis is a or somatic apospore, we may call it. It is a very common in some certain species of gymnosperms, angiosperms, but never found in gymnosperms. So it is advanced character. तेणु आमर पॉली apomixis गोटे advanced character कहुच्छी कारण gymnosperm ना और apomixis तुम देखा सबू प्रकार फैमिली ना. When you observe different plants, we find 40% of apomictic characters plants are from three families. One is your Asteraceae, other is your Poaceae, other is Rutaceae. ये तीनटा फैमिली बेसी देखा जाए कौन apomixis जोठी कि तमरा विदाउट फर्टिलाइजेशन सीड रे एम्ब्रियो फॉर्मेशन होची 40%. किंतु टोटाल जदि हम देखा नंबर ऑफ स्पेसीज इवन लेस देन 10%. 10% रू कम गछ एतिर अछि. सो व्हाई इट इज सो दैट्स व्हाई द रीजन इज सम apomictic character is genetic. बट हमर एई फैमिली गुडा करे तांकर जो जीन रे किसी अपोमिक्टिक कैरेक्टर अच्छी सो दैट सेठी बेसी देखा अन्य कहीं देखा कम देखा दैट्स दिस थ्री फैमिली आर डोमिनेंट सो व्हाट इज द रीजन मेकानिज्म कौन आप कहीं हो सो लेट अस डिस्कस द रीजन ऑफ अपोमिक्सिस फर्स्ट थिंग इज द one cell any cell can develop into the new plant as per the totipotency theory the Haberland जो कहते हैं according to Haberland any cell of the plant or animal having a complete set of genome can develop into a new plant suppose similarly complete माने कौन आमर तो diploid cell अच्छी गोटे 2n number of chromosome जदि haploid cell आम आनले n number of chromosome गोटे pollen गोटे आंथर आनले आंथर जो आंथर haploid cell से haploid cell में haploid plant हो पारिबो haploid plants can be produced that's why because having one set of genome so when a complete set genome रहिबो जदि सेटा को आम under certain conditions we can grow into a new plant or new animal that we call totipotency so this phenomenon is there but why sometimes that happens in plants सब बेले कहीं होनी not always not in all plants the reason is the generation of the cell capable of forming the embryo without prior mitosis is not available in all plants so the capacity first thing is the capacity of a cell to develop into embryo without fertilization that must be there the cell must be having the capacity all cells are not capable सबू सेल कैपेबिलिटी न थाए तो आम कहे सब सेल हो पार कि दे आर नॉट कैपेबल तुम आम जो टिश्यू कल्चर कर गोइंग टिश्यू फ्रॉम सेल्स सो वी पर्टिकुलर प्लांट वी टेक पर्टिकुलर टाइप ऑफ टिश्यू वी सम प्लांट्स वी गेट लीफ टिश्यू some plants we get root tissue some plants we get mesophyll tissue some different tissue some plants we get pollens so different tissues have different types of regeneration characters तो जहाँ regeneration character बेसी थी जो सेल से ही ताकू से एम्ब्रियो हो पार दैट्स व्हाई the generation of this so the capacity of the cell to develop the genetic capacity we can call it then your fertilization is spontaneous independent development of embryo without fertilization second is independent development without fertilization usually what happens after fertilization this zygote develops into embryo fertilization होला पर zygote embryo develop करियो करेक्ट सेटा ताको स्टिमुलेट करियो the fertilization process stimulates zygote develop but this cell if it has the independent development without embryo capacity हो पारिबो ताले सेटा तमरो embryo करि पारिबो so even if there is no fertilization if this cell is capable of developing into an embryo then it can produce embryo if this is not capable you will try it will not produce the embryo then your sometimes automatically endosperm is also produced either this when the embryo is produced the endosperm is required because embryo जो produce होगा तब खादी का endosperm था so when there is no fertilization there is no endosperm so either this vegetative embryo without fertilization this embryo may develop stimulate endosperm formation endosperm formation भी करिवो हबो किंबा endosperm सेठी तयारी हेतिबो so either it will the endosperm will developed or it will regenerate so endosperm regeneration stem formation so either endosperm will be formed जदि सेठी सपोज एम्ब्रियो फॉर्मेशन है नहीं फर्टिलाइजेशन है जाइगोट है नहीं किंतु यदि एंडोस्पर्म फॉर्मेशन है तो एंडोस्पर्म टिश्यू अच्छी सो एंडोस्पर्म आइदर इट विल फॉर्म और दिस एम्ब्रियो will stimulate the cell which developed embryo it will stimulate the formation of endosperm so that you will get the fruit so these are the basic things required to for apomixis to produce a viable embryo in a viable seed even there is no fertilization जो नहीं there is no syngamy what you calling syngamy the fusion of male and female gametes there is no fusion but the embryo can be produced and if the cell is viable and the cell can develop to without fertilization and the cell can also produce endosperm or if endosperm is there it can use it endosperm sangre tar link nahi कारण endosperm ta sangre कार embryo रे link अछि जोटी तमरा zygote ro because during the fertilization the zygote will develop in the embryo and the triple fusion tissue triploid tissue from the endosperm but when there is no fertilization there is no endosperm so when there is fertilization triple fusion but no zygote then it is okay because endosperm will form but when there is no fertilization there is no endosperm this should have the capacity this embryo should have capacity to generate this endosperm like tissue so that it will provide food so this is about the formation of the mechanism by which the aposporic embryos are formed then What are different types of apostrophe letters and pathway? Now in normal pathway, what happens? This seed, when it is formed, if this is the, suppose this is the ovary, this inside this is the ovule, inside this there is the nucellus, then this is the integument, here is the megaspore mother cell. This megaspore mother cell will divide into megaspores, and this megaspore will develop into the embryo sac. This is the, suppose this is the megaspore mother cell, MMC. This megaspore mother cell will undergo meiosis, forming 4 haploid megaspore, out of which 3 degenerate, 1 remains to form the embryo sac, which is 80 nucleate normally. One egg, 2 synogytes. This is the normal process. Then after this formation of this embryo sac, the fusion occurs, gametic fusion occurs, where the one pollen, one sperm cell fusses with the egg, and other fusses with the secondary nucleus, from the triploid nucleus. This is normal pathway. Then embryo formation is there. Then this is from the embryo. This is the endosperm. The normal process. But here in case, what happens? In case of apomixis, these stages are not there. So when the cycle starts, apomixis, this fusion, this fertilization is not there. So there is direct formation of embryo. From this megaspore mother cell, even for megaspore, direct formation of embryo. This will bypass. এটা କ'ଣ ହେଉଛି ନା ବାଇପାସ୍ କରିଯାଉଛି କେଉଁଠି ତୁମର ଯେତେବେଳେ ଯେଉଁ ଏଗ୍ ଫର୍ମେସନ୍ ହେଉଛି ଆଉ ଫର୍ଟିଲାଇଜେସନ୍ ହେଉଛି ଏଠି ବାଇପାସ୍ କରିଯାଉଛି ସିଧାସଳଖ ଏମ୍ବ୍ରିଓ ଫର୍ମେସନ୍ ସେ ସିଡ଼ିରେ କରୁଛି ସୋ ଇଟ୍ ମେ ବି different types. Sometimes it may be your sexual seed formation, or it may be when we study details. Sometimes we find diplospore is there. Diplospore means here is case that direct megaspore mother cell, it becomes diploid. Diploid megaspore. This diploid megaspore directly develop into the diploid embryo. So there is no formation of egg and there is no fusion of gametes. And sometimes there is apospory. Apospory means this spore, this megaspore, it may form the embryo. And sometimes you will find the parthenocarpy. Parthenocarpy happens when the other cell, here not the megaspore mother cell is not there, it will not function. Other cells of the gametophyte sporophyte, it will form into the by parthenocarpy, parthenogenesis from the embryo. So the formation of embryo, normally it is the your sexual embryo. Here the sexual embryo. But without sexual fusion, it may be due to diplospory, it may be due to apospory, or may be due to parthenogenesis. Parthenogenesis means what happens? The development of the any cell into embryo without fertilization. And in parthenocarpy also there is term you have to seed, formation of fruit. Fruit without fertilization. Sometimes when there is no fertilization, means fruits are formed. There was a papaya, grape, there is no seed. When we develop the fruit, there is no seed, without seed, that is sometimes commercially very important. ତା ଭିତରେ ମଞ୍ଜି ନାହିଁ ମାନେ ଅଧିକା ଦାମ ସେଇଟା. So seeds are not there, fruit is there. So because sometimes seed create problem. ଆମର seedଟା ଦରକାର ଆମେ ଗଛ କରିବା ପାଇଁ, ଯଦି ଆମେ ଖାଇବା ପାଇଁ ସେଇଟା ଅସୁବିଧା କରୁଛି. So if you get a fruit without the seed, suppose there is no cell inside the mango, ଆମ୍ବ ଭିତରେ ଟାକୁଆ ନଥିବ, ଖାଲି ଆମ୍ବଟା ଥିବ, how tasty it is. So now scientists in China are trying to develop it. There is no embryo, no seed, only fruit. So that is the advanced development. Now we find in case of banana, in banana there is no seed, natural seed. କଦଳୀ ଭିତରେ ମଞ୍ଜି ନଥାଏ, ଯଦି ଯେଉଁ ଟିକ୍ ଟିକ୍ ଥିବ ସେଇଟା ଗଛ ହେବନି. So they are natural parthenocarpy. So this fruit is formed without fertilization, or there is no seed. But in case of parthenogenesis, sometimes embryo is formed. Fruit is formed parthenocarpy. The ovary develop into fruit, but ovule develop into seed. So when we also help develop the ovule, there is no fertilization into the seed forming embryo. So the ovule टा develop कले आमे तादरे सीड फॉर्मेशन कला फर्टिलाइजर नईकरी भी से ओव्यूल सेल एम्ब्रियो बाहरला सेटा आम कह पार्थेनोजेनेटिकली डेवलप्ड सीड. बट व्हेन वी डेवलप ए फ्रूट विदाउट सीड, दैट वी कॉल पार्थेनोकार्पिक सीड. दैट सेम थिंग बट डिफरेंट पार्ट इज स्टिमुलेटेड. आम ताको कौन करछे ना हार्मोन देबा डिफरेंट मेकानिज्म रे बा डिफरेंट जेनेटिक तार म्यूटेंट अच्छी जोड़ा सीड हम नहीं. पपया तुम देखो मार्केट जो अमृतभंडा सब बड़ बड़ पपया मिलूँ, दैट इज नो सीड इनसाइड, because they are used by the parthenocarpic mechanism, parthenocarpic process. They have developed those plants, but this plant cannot be grown. सेटा कौन सीड आ तादर फॉर्मेशन हेनी. सीड फॉर्मेशन वाला कौन मतलब से प्लांट पाइबानी. जदि आम गोटे ग्रेप्र गोटे अंगूर मंजी ना आम कौटू करवा ताले अंगूर डालू काटी कर किंतु पपयार धर पपया सीड ना आम पपया डाल काटी कर. सो वी नीड फॉर दैट केस कदली तो मंजी यदि होनी आम कौन काटी कर. सो इन दैट केस वी नीड आर्टिफिशियल mechanism to develop vegetative cell into the new plant, that by tissue culture. So now this in case of this apomixis, the different methods, it is very useful also, but the advantages are there. The apomixis is used in various fields of agriculture and agricultural research and studies. So apomictic seeds are normally what happens when farmers get the seed, we get the seed hybridization, hybrid seeds. Hybrid seeds after 1 or 2 generation they segregate. So when we develop a hybrid plant, जदि आम गोटे हाइब्रिड राइस सपोज कले हाइब्रिड राइस गोटे दुईटा भेराइटी आनिक नुआ राइस कले आम जब बुणवा किसी जेनेरेशन पर से क्यारेक्टर रही फास्ट जेनेरेशन रही एफान क्यारेक्टर एफान जेनेरेशन वी कॉल इट आफ्टर 2-3 years दे विल बी सेग्रीगेशन देयर विल डिफरेंट टाइप्स ऑफ प्लांट्स ऑल द ओल्ड क्यारेक्टर्स कम आउट. सपोज वी आर गेटिंग वन आम जब गोटे दुईटा गच हाइब्रिड करचे गोटे हूँ तुम डेंगा गच से तुम अधिका दिन पर फल हो छोट छोट धान हो गोटे छोट गच कि कम दिन में फल होम हाइब्रिड कले. सो दैट वी गेट ए स्मॉल प्लांट मोर नंबर ऑफ सीड्स इन ए वेरी शॉर्ट टाइम कम दिन में बहुत फल पाऊँ. जदि आम बुणवा दिन बारंबार से मंजी रखा कौन सेग्रीगेशन आफ्टर मैनेजन इल फाइंड से डेंगा गच भी थी गेड़ा गच थी बड़ केना थी छोट केना भी थी से कौन मैंने यूजलेस हो. दैट्स वाई फार्मर्स दे नीड ए सीड एवरी ईयर because segregation. And when we send the market आमर hybridization करुचे we are making the hybrid because of apomixis or because of artificial hybridization process आम seed तैयारी करुचे we send it to market. But after before sending market we try it several times. आम ताको ट्राए करके देखु से कौन विभिन्न जब टेस्ट कराऊँ गोटे परीक्षा जमी बारंबार कराऊँ भल पिला बाछा सेमती कौन बारंबार आफ्टर 2-3 generations और 5 generations we find the suitable जोटा कि सेग्रीगेशन होनी बाछी बाछी आम मार्केट को पठाऊँ. So that they are commercially they are good seeds. But in case of apomixis what happens? The seed there is no seed. So when the seed is there is no fertilization. तेणु कौन ना आमको डाई हैप्लॉइड जो गोटे सेल. So there are different benefits or applications we call. So applications of apomixis. So most important application is your economical seeds. That means seeds are cheaper. When we hybridize, आम जो गोटे हाइब्रिड सीड तैयारी करवा आमको गोटे टाइम लगे और बहुत दिन लगे. आम गोटे धर 2टा भेराइटी गोटे हाइब्रिड कर नुआ भेराइटी बाहर कल तार मंजी बुणला से कौन गच हम देखा पड़ो. आम जो गोटे सपोज गोटे बानाना भेराइटी बाहर कले आम से 2 वर्ष पर जाना प्रकृति से कौ कदली हो तार कदली फल से प्रकृति में जमती हो कि अलग प्रकार हो कि राइस आम हाइब्रिड कले आम 10टा 20टा सीड हाइब्रिड करचे से कोटा बुणिकी देखिले जब गच है धर आम 500,000 गच पाइबा नेक्स्ट जेनेरेशन वी टेक टाइम एंड इट कॉस्टली ऑल्सो से हाइब्रिड सीटा दाम बेसी. बट हियर व्हाट हैपेंस इन इकोनॉमिकल बिकॉज डायरेक्टली आम जो apomixis रे सीड पाउचे सेतरे किछि हाइब्रिडाइजेशन नाही द सीड्स आर वेरी प्योर बिकॉज ओनली वन सेल डेवलप्ड इन द एम्ब्रियो जदि गोटे सेल रु एम्ब्रियो होउची तेणु कोन होउची ना से सेल रे किछि इम्प्यूरिटी नाही जहाँ जीन अच्छी दे आर ऑल प्योर सीड्स दिस प्योर सीड्स दे आर वेरी यूजफुल इन द एग्रीकल्चरल पर्पसेस सो चीपर ऑल्सो एंड देयर इज नो सेग्रीगेशन आम से तार किसी कन्फ्यूजन हेनी आफ्टर जिते जेनेरेशन आम से बुणुवा तार नुआ कैरेक्टर किसी बाहर कारण से कैरेक्टर प्योर अच्छी देयर इज नो हाइब्रिडाइजेशन ऑल्सो नेचुरल सीड्स सो देन इट इज hybrid development is shorter and easier. Why? Because here when we get the seed, because the apomixis we get the seed, it is 2n, it is pure. So one variety 1 and variety 2. We make the hybrid, we get very pure seeds. कारण से तार मिक्स कैरेक्टर ना आम जो दुईटा गचर हाइब्रिड करुचे आधे भी कैरेक्टर किसी मिसामिशी रही ताधे मिसामिशी रही तो आम जो आणुचे केते दूर सबुजा कैरेक्टर थी आम जापर आम अपेक्षा करा पड़ो वी हैव टू फाइंड आउट सो हियर इट इज इजियर बिकज वी नो दिस इज अ प्योर सीड दिस इज अ प्योर सीड सो वी नो दैट सो देयर इज नो adulteration so when we get the seed after hybridization we get it easier and development is very quicker also so it is shorter and easier way of hybridization for hybrid plant breeders it is the best technology. Then it is here there is some cases of male sterility. It is independent of male sterility. Male sterility means in some cases these plants produce egg, but the pollens are sterile. These pollens cannot produce seeds. Both in case of many cases of palm trees. Palm tree रे कौन हम सतार जब सीड थे हो पारे फॉर्मेशन हम सत बिकॉज द स्टेरिलिटी दिस मेल स्टेरिलिटी बट हियर वी डोंट नीड एनी मेल pollen or male sperm cells only one cell that develop diploid cell that develop करछि सेठी हमर मेल और फीमेल से दरकार ही नाही we don't need male and female separately so even if there is a male sterility if the plant is male sterile suppose we get a plant that plant is male sterile we cannot get the pollens viable pollens in that case we could not get कारण आमर जो कौन डबल कोकोनट जो प्लांट अच्छी दैट इज ओनली वन इन द इंडिया बॉटनिकल गार्डन in that case what happens in that case the plant is male sterile सेठी तार फ्लावर होउची किंतु से तार pollen टा सेठी से गचर तार स्टेराइल pollen से तार फर्टिलाइजेशन हबो नै सेटा हम कोन करबा ना आमे सेठी साइंटिस्ट माने बाहर जाइकरी अन्य कंट्री रु जाइकरी हमर आइलैंड रु नाइकरी pollen आनिकरी ताकू you took them 4 years to develop the seed but if there is no need जदि सेठी आमे डायरेक्टली apomixis जो सीड आमे करि परिबा we can directly say so there is no need to get the pollen find the pollen somewhere else so that is called the male sterility so there is no need and then independent of male system pollination yeah independent of pollination for is in case of hybridization the one important part is to collect the pollen item and put it for breeding but in case of apomixis when we get the seed directly from the vegetative tissue so there is no pollination so pollination is required so there is no need of pollination so in case of developing seeds by the process of apomixis there is no need of pollination so it is independent of pollination किसी दरकार ही नहीं that part is not necessary then your survival interspecific crossings when we do survival more survival usually what happens when we make a cross between two different varieties suppose in two different species interspecific we are calling so there is a rate of survival very less जदि कहना तार दुईटा प्रकार जो स्पेसीज अलग अलग स्पेसीज तांक भितर कंपेरेटिव वी कैन गेट फर्टिलाइज सीड द फर्टिलाइज बट सीड व्हाट हैपेंस द सीड विल नॉट सर्वाइव द एम्ब्रियो विल नॉट सर्वाइव इट विल डाई सो दिस इज कॉल्ड द सर्वाइवल ऑफ इंटरस्पेसिफिक हाइब्रिड सेठी मरीଯିव गछटा बंचबनी बुझाले बंचेनी एम्ब्रियोटा मरीजे एम्ब्रियो बिकम्स abortive but in case of apomixis so there is no problem because when the by apomictic embryo when we get the fertilized or get the seed zygote by seed by fertilization in the next generation there is no problem because there is no so interspecific hybrid crosses survivability problem haboni kana seta bonchi paribo it is already the seed embryo is having this character टू सर्वाइव एडवर्स कंडीशन जो डिप्लॉयड सेलटा जो एम्ब्रियो फॉर्मेशन करी एडवर्स कंडीशन से तार सर्वाइव करें एडवर्स कंडीशन सो दैट इट इज मोर देन योर डिजीज डिमिनिशिंग डिजीज. So when in case of normal formation of normal seed, we get the pollen from one plant, we get the egg from other plant, there is a zygote. So in that during transfer of these vegetative pollens from one to other, we find the zygotes. Zygote contain many many viruses which cause a very minute structure. They come with the zygote, they stay with the zygote and they produce the disease. The disease continues. But in case of apomixis, there is no foreign body. So foreign body is not coming. That's why it is free from the any viral disease. So when we get the seed from the apomictic mechanism, we get the viral free disease free seeds. This is about the application of polyembryony and apomixis. Thank you.